So, dude, like we talked about many, like, dude, like you guys already know what DNA is, right? Like DNAs and RNAs. But keep in mind, guys, like they didn't. I mean, they knew, the existence of DNA, deoxyribonucleic acid, but they didn't know this was something that's inheritable from one to the other, from the parents to the offspring. They didn't know what it was. They didn't know it was a protein or DNA. They had no clue. So a lot of times they thought it was a protein that's coming down from the parents to the offsprings. Okay, but I think you might also have to know some of the scientists' name here, by the way. So I think like what was that, like Avery? I mean, this is where you got to do the reading, okay? Because I don't memorize the whole like the scientist's name. So Avery and Griffith. Okay. So they did some testing. Okay. So Avery, McCoy, or Griffith. Well, there are three of them. Okay. I mean, I have no offense for the other one, the last one, like McCoy, whatever. Okay. So they did some testing on, okay, so what is inheritable, down to the offspring. But so basically what they did is, okay, so this is where the reading comes down to, okay. So they have a pathogenic prokaryotic bacteria, okay. So pathogenic means something that cause a disease, okay. So they put this pathogenic prokaryotic cells into a mouse, okay. They gave this to a mouse, and of course the mouse dies, okay. The mouse dies because they put a pathogenic bacteria there, okay. And then, so they did a control probably. This is what they call a negative control. Negative control means something doesn't work, right? We talked about the control and stuff like that, right? So they put a non-pathogenic, okay? So it's the same species of the prokaryote, okay, but non-pathogenic, okay? Prokaryotic cell into the mouse, okay? The mouse survived, okay? So like, yeah, they're alive. I mean, there's like, okay, so there's nothing happens to the guy. Of course, like it's not pathogenic, right? So basically, so this is the pathogenic. So I'm gonna put that as a red one. So this is what they did, okay? So next thing that they did, I mean, step by step, right? Like you can do the reading if you want to. They actually heat up. So they heat it, okay? They heated the pathogenic bacteria, okay? And then whatever that was heated in, whatever that was heated, okay? They feed this into the mouse, okay? And the mouse was what? They survived, okay? So when they heat it up, it's okay, okay? And now the next thing they did, okay, is that, okay, so they, whatever that was heated, like, when they heat it up, the what do you call that, guys? Like the proteins goes away, like blah, blah, blah. You guys know the whole drill here, guys, by the way. So the next one, so how these prokaryotic cells are pathogenic, I mean, it could be many things, okay? They could produce toxic maybe, so they got some DNAs in there, they can produce some toxins, and, that's the reason why they might die. Who the hell knows, okay? So what they did is this, okay? The heated pathogenic prokaryotic cells, like they're dead, okay? And then they put those, so basically what they did is, okay, so whatever the dead particles of the pathogenic prokaryotic cell, they put these two together, okay? So this is a non-pathogenic, okay? And that's the leftover, for the pathogenic prokaryotic cell. They left those, they put those together, okay? And then they put them into, or the concoction of what this is, okay? They put them into a mouse, okay? What happened? The mouse died. Okay, so again, the significance of these guys are what? Okay, so something that was the deadly material, whatever that is, okay, whatever the dead material of the pathogenic bacteria, whatever that was, somehow got into the prokaryotic cell, okay, and then it killed the mouse. Okay, so they still weren't sure. Okay, it's like, dude, so what went in there? Is a question, okay? I mean, now, like, we should know now, what it is, okay? It's what? Probably what? The DNA, okay, which is left over from the pathogenic prokaryotic cell, went into a non-pathogenic, and what made them pathogenic, okay? And then it's gone in there. So they actually asked this question on the AP as well. Like AP, like this is last time the AP. So so one of the things you gotta understand, guys, like, so just between us, right? Like, Like, why are they taking the, like, so, you know, like, outside of these guys, they didn't say this, because they're, like, very primitive at the moment. They didn't really know a lot of DNAs and stuff. So, this is a non-pathogenic prokaryotic cell, right? Why would they take an outside DNA? Why would they take them in? That's a question to ask yourself. Like, so it's like, okay, so why are they taking a random DNA in from the surroundings, right? You gotta know the name of this, guys, by the way. It's called transformation. The process is called transformation. So transformation is, okay, these prokaryotic cells uptaking of a random DNA in the surroundings. Okay, that's like usually like how they just pick up a random DNA. Okay, so the reason for that is these prokaryotic cells never go into, okay, like when they reproduce, a lot of times they go reproduction in terms of what? Binary fission. Okay, they actually divide, right, by themselves. So the problem with this, guys, by the way, I mean, so we're not really talking about the reproduction right now, guys, by the way, but the reproduction that we have and other, you know, like, you know, reproduction that we go through, right, versus what they go through. So their reproduction, which is what they call asexual reproduction, right, because it's like they replicate their DNA and then just get it over with. But the problem with that is what? Every one of these guys will be exactly the same, okay, as what? Like, yeah, itself, right? So the problem with that is what? Evolution perspective. I mean, so yeah, something can kill one of these guys, can wipe out the entire population. Okay, so it's the nature of the entropy. Okay, where what they want to have what a greater variety, a greater what entropy. So, which means that's what led to what? I mean, so you know, history perspective. That's what led to what? Make them take in all the what? Random DNAs. So basically, guys, means that's what transformation is. There are three ways, okay? The better terminology is recombination of DNA, okay? So three ways the prokaryotic cells can recombine their DNA, okay? Is transformation, transduction, and conjugation. So three ways. I'm not like this is a chapter, you know, something that we'll discuss probably next week. I'm just saying. So basically, how this occurred. Because, well, yeah, they uptook a DNA. But they still not sure, okay, was it a protein or DNA? They wasn't sure, they weren't like too sure about this. And the next guy, okay, the Hershey and Chase, okay? Dude, this is all like late 1800s and early 1900s. Like, it's like pretty recent. Like, dude, like you guys were born like what, like 2000 something, you know? It's only 100 years ago. Okay, so you know, like, dude, only 100 years ago. Like, dude, like how long has been like the existence of living things, guys, by the way? Like what, billions of years? This is only the past 100 years, okay? So this is pretty recent. So you know, next folks, guys, you make sure you guys do your reading, okay? Is the Hershey and Chase, okay? I'm not gonna write the name. So you probably heard this, okay? Hershey and Chase. So they use something they call phage. Okay, so phage, guys, is, by the way, is a virus. Okay, it's a bacteriophage. Okay, it's a virus. Okay, so like, dude, I gotta tell you guys, okay, every single day, okay, just so you know, 50% of the prokaryotic cells die due to attack from a virus. They die, dude. They just like, you know how the virus work, right? They inject their DNA and what kill what the prokaryotic cells or whatever cell they can hook onto. So these guys can easily kill. I tell you guys, like if you have thousands of the prokaryotic cells in a petri dish, when you introduce some viruses, guys, it can wipe out the entire thing in like 5 minutes. So they're very lethal, by the way. They're very lethal. They're bound to kill all the prokaryotic cells. I mean, so like you know the COVID, like dude, it's crazy, right? Like it's stopping like people from, I don't know. It's virus is very lethal, by the way. Okay. So if you think about it, right? So basically, after what they found out, because virus is very simple, like it's not an organism, it's a simple thing, right? So they have a protein capsid, okay? So again, I'm gonna drug you very simple stuff, okay? Protein capsid, and they have a DNA. Okay, it could be a DNA, it could be the RNA, it depends, right? So basically what they did is they use a radioactive material. So as we know, so DNA, okay, they have what, guys? So they found like elements that are present only in DNA, they're only present in the protein. So protein as well, protein's got sulfur. Sulfur is unique to a protein. There's no sulfur in DNA. We talked about this, right? So again, you guys should know which amino acid about sulfur. We talked about it. There's sulfur in what? There's a what? Methionine? Methionine? What's that? What has sulfur, guys? That's amino acid. You recall, guys? What has sulfur, guys? There is something that makes a disulfide bridge. I forgot.
Cysteine?
Huh? Oh, cysteine. Yeah, cysteine. Cysteine is one of them, right? And there's one other one, methionine. Okay, I think they're the only ones that have sulfur, by the way. So, you know, so these are stuff that you have to memorize. Okay, so the other ones don't have it, right? So they use a radioactive sulfur, okay, on those two amino acids. You gotta know them. AP, you don't have to. I mean, if you know it, like, you write it down. But I don't think you really have to know. And also, what, guys? Like these DNA, they what? Phosphorus. So they radioactively label the phosphorus and sulfur on the proteins versus the DNA. Okay, and I mean, so like, dude, you gotta understand, right? This is what, prokaryote bacteria, right? Dude, whatever goes in, so this is what they're thinking, okay? Whatever went in, okay, killed this cell, okay, and what? Replicated this guy. You understand what I'm saying? So, which means what? Like, yo, dude, they mass produce the same virus, okay, after these guys go in and attack them. So, what they're thinking is, whatever that's injected to the prokaryotic cell is what? That's what they call, it could be what? Inheritable material, okay? So, is it a protein or what? A DNA, that's the question they've been posing, okay? So what was it, guys? Of course, what? They found what? Whatever that went in is what? The radioactive phosphorus. So they were able to find what? Radioactive phosphorus inside the prokaryote cell, and what? The sulfur was always what? Outside. So that kind of solidified what? Okay, it's what? It is the DNA. It's the DNA that what? Ultimately will be handed down to the next offspring. So they were skeptical about the DNA being, inheritable, because it's very simple structure for them, because it's a repeated unit of ATGC. Okay, so it's repeated. So like the structure is so simple. I mean the whatever that's in it is not so complicated compared to a protein where what? It's like there could be millions of different proteins out there. So they thought the protein was a better option compared to what? A DNA, but they just found that, well, like based on based on the experiment that they did, which means they found out, well, okay, so it is what? The DNA. So now there's a massive research based on the DNA, the structures and all that is being, like from here is massively invested now. So that's where you probably heard the terms, the folks guys like Watson and Crick and all. That's when, they start to find out, well, it's DNA. So. In regards to the structure of DNA, which is nucleotide basis, guys, I mean, so that's all the stuff that we talked about. I'm not going to waste too much time there. So, like, there's a different guy called, you guys have to read it, guys, like Chargaff rule. So, so this is all, like, before, like, they were able to find the structure of the DNA. Chargaff says what this guy said, what, okay, so there's a certain rule, okay, that this guy said, okay, so adenine, okay, and the thymine, the amount of abundance in the DNA, they're about the same. C and G, they're about the same. So Chargaff is the guy that said, well, okay, so A and T, the amount in the DNA is about the same, and C and G is about the same. So that's like they said, okay. So it's obvious. I mean, it's obvious stuff, right? But all these scientists contributed for what? For the structure that we have right now, okay. So nothing fancy, but dude, like structure. Watson and Crick, I mean, they're not going to ask for the structure for the Watson and Crick for the DNA structure. It's a helical, it's a double-stranded, like you probably heard that, guys. I mean, I'm not going to go too crazy right now. But you do have to know, I mean, so we definitely talked about it, guys, like pyridines and purines, so which is, you got to understand, guys, like A and G, these are what we call purines. And then we have a C and C, U, and then T, these are what? Pyridines, right? So we talked about it, guys. This is what, like one single carbon ring. Well, this is what? This has a 2 carbon ring. Okay, we talked. This is actually bigger than this guy. We talked about that, guys. By the way, so based on, like, I mean, you can do the reading, guys. By the way, so based on what they found out, okay, based on the radius or like the diameter of the DNA helical shape, okay, so they can only fit, okay, or it makes sense where what you should have 1 purine and what 1 pyrimidine. Okay, so that's this is the diameter. Okay, so only time the diameter makes sense is what? Like you have one of this guy, one of that guy. So that's how they figured it out. So which absolutely makes sense because what? Chargaff said, okay, so there's what? About the same ratio of A and T, okay, same ratio of what? C and G. So which means, well, yeah, like they always get paired together, and that's basically what they found out based on the chart diagram rule, just so you know. And I do, it's a very interesting stuff that you can, if you do the reading guides, just so you know. Okay. They also, we know the basis, how it looks like, right? So we have a phosphate, you know, and then there's a carbon group here, and then there's like nitrogen space. So, I mean, you should kind of know, guys, what it looks like, okay? So we know that, you know, let's look at it. So I gotta draw this a little backward. So this is, so basically what I mean, in terms of, if you look at it, in terms of the DNA, so they call this, guys, we talked about it, guys, one is double-stranded, the DNA is double-stranded, the one goes from 5 to a 3 end, the other one is what, guys? It's what they call as anti-parallel, so the other one is what, from the other 5 to a 3 end. So if you look at the DNA, this is what, like 5 to a 3, this is a 5 to 3, so they're anti-parallel, that's what we say, okay? So keep in mind, guys, like they talk about a lot of these 5 and 3 stuff, okay? And we talk about why they're called 5 and 3 is what? This one is what? If you look at it, this is the carbon number 1, 2, like 3, like 4, and 5, okay? There's a 5th one here. So basically what happens is, it starts from the 5 and what? There's another one hooked up on the 3rd carbon. So that's why I call it 5 to 3 end. That's 5 and 3 comes from what? Number of carbon that we talked about, okay? On the ribose. So you get, we call it deoxyribose, ribose. We talked about all that stuff, so I'm not going to repeat that. So without like wasting my time, guys, I mean, so basically, you know, like you probably heard Rosalind Franklin and Watson and Crick, they're all about the structure of the DNA. That's what they got the Nobel Prize for, okay, by the way. So which is, they don't ask you about the structure of DNA. So they do ask you about the DNA replication. So let's talk about, guys, the DNA replication process. So make sure you do your reading, guys, okay? So I'm sure, guys, like whatever the bio you took, I mean, I'm sure you heard about DNA replication, right? So DNA replication. Replication, I'm sorry. So this happens where, guys? In a cell cycle. Okay, we talked about cell cycle as just as G1. This is S. This is G2. This is the M phase and C phase, right? Where does this occur, guys? It happens what? At the S phase. It's called a, you know, what we call the synthesis. It's what? DNA synthesis. Okay. So I mean, like, dude, take a look at this, okay? So we have a strand of DNA, okay, a long piece of DNA. So we got 46 of these guys, okay? So we have this is a 5 to a 3 end, okay? Our DNA is what, guys? As a complementary strength, okay? And I'm sure you heard the term, guys, by the way. Replication of DNA is what? They call this what? Semi-conservative. They call this what? Semi-conservative, okay? So, like, okay, so very simple way to explain this, okay? Semi-conservative means what? So when this is the DNA, right? When they make the copy, right? Like after this, what it looks like is what? So it's not like they're getting a brand new DNA, right? What happens then? 2 old strands are what? Separated, and what? The new strands are what? New strands get attached on them. So that's what they call semi-conservatism of the replication of DNA. So what I'm saying is, guys, if it's a fully conserved, what happens, guys? Yeah, dude, this is what you get, right? You leave this alone, okay, and what? You create what? Brand new one, okay? That's what that's fully conservative. That's not what it is. So these guys will separate, and what? It will have what? New strands of DNA attached on both of them. So that's what they call semi-conservatism. Which is like this is something you should probably have learned in your like whatever 7th grade bio or something, whatever you learned. We'll get to get into a little more details, guys. Okay, so even now, guys, right now, this is like AP stuff. Okay. So one thing to note, guys, I mean, so on a DNA, okay, there are multiple origin of replication. So there's like few spots here. Okay, that what can be considered what? It's what they call the origin of replication. So what will happen is, it's a textbook reading, guys, will explain a lot better, okay? So I'm gonna only draw like one of them, okay? So I'm not gonna draw like many of these guys, okay? So what will happen is, guys, there will be a helicase. Okay, there's a helicase. Will be running. So the first thing first is what? There's a first one is a helicase. Helicase unwinds the DNA. So unwinding is what? Basically they're prying them then open. They pry them open, breaking the hydrogen bonding. Okay. The helicase will run down, okay, to one side of the DNA, and it happens on both sides, by the way. So they open up like on both ways. Okay. They start from a single point and open up on both sides. So basically, what will happen is they create something they call, guys, a replication bubble. So it kind of looks like this. And that's what it looks like, guys, by the way. So which means there's a helicase running down, okay, unwinding them. Okay. And next one, guys, and again, this is something, again, like something, if you read it, guys, it will definitely help, guys. Okay. Single-stranded binding protein. Okay, it's called single-strand binding protein. Okay, it's called the binding protein. Okay. So basically what it does is what? They make sure that whatever this thing is, okay, they make sure what? The two strands that are separated by the helicase are not what? They're not sticking back together, okay? They're not like, okay, it's coming back together. So that's what they call, guys, a single-stranded binding protein. Keep in mind, like, which means, well, yeah, keep them what? Single-stranded. And now this is where, guys, okay, this is where, guys, like, students are getting a little struggle, okay? Like struggle a little bit. So they don't draw like this. Okay, a lot of times they draw like this. Okay, so let's kind of not look at this part. Let's not look at this part. Okay. So I'm gonna just like break this in half. A lot of the textbook, guys, like this is only half they give you. Okay. So right now, guys, this is the 3 end. Okay, this is a 5 end right now. Okay. So after these two, okay, so this is where, guys, it gets a little interesting now. Okay. So I'm sure you heard of a guy called DNA polymerase. I'm sure you heard that guy. Okay, that's the guy that goes in and what, like start to match the pairs, right? So let's take a look at this guy. Okay, so that guy's not gonna come after these two. Okay, the next guy is not this guy. The next guy is what? Actually called primase. Okay, it's called a primase. You gotta know it, guys, even for the AP. So the reason is, okay. So this is kind of funny because DNA polymerase, okay, they're only good at what? Extending an existing piece of strand. So which means, okay, so their job is not to initiate, okay, the matching process, but what? Elongate the matching process. Okay, so so it's like, okay, so they can't just like go and what? Just like start replicating. It's what? They need an initial piece in there. And what? Extend it. So that's the reason why they need what? A primase to do so. Okay. So primase, guys, just wait. That's what will happen is primase. They will be start to put. Okay. They start it off. Okay, from the 5 to a 3 end. Okay. So they put a primer there. Okay. So they put a primer there. And then and then start to. And then what happens, guys? And then what will happen is the DNA polymerase are now going to be able to extend this piece. So the problem with this will be a big issue, guys. This is something that we talked about, guys, like telomere and that stuff, guys, by the way. So the reason for that is a primase, okay, the basis they put is not really what? Deoxyribonucleic acid. Like it's not a nucleotide DNA, it's not a DNA base, what? They put an RNA base in there. So they do not put deoxyribose. They put the one with the ribose, which is what? They use that for the RNA. So look at what, guys? This piece is what? An RNA piece, not a DNA piece, okay? So which means what? Later they have to take this guy out, okay? And what happens, guys? So yeah, now after primase, what happens, guys? And then DNA polymerase, now they're in action. So which means what? Yeah, this as they unwind, what happens, guys? The DNA polymerase will run down and what? Extend a piece of DNA now, okay? So keep in mind, guys, like listen very carefully, okay? DNA polymerase, they can only produce the DNA from 5 to 3 end, okay? They will be only produce the DNA from the 5 to 3 end, okay? So they have to run down from what? Existing piece, which is from 3 to 5, okay? So existing piece that they're copying, okay, they're gonna run down from 3 to 5 end, okay, and then by what? And then matching what? And then creating a strand there of what? Complementary strand of 5 to 3 direction, okay? So they can only produce 5 to 3 direction, okay, make sure you understand. So in your AP, this is it, okay? Your question, that's all they ask you, okay? You get to know a little more for the Olympiad, okay? But it's not in the reading. Why is it 5 to 3 end? That's the question they ask you. Why is 5 to 3? Your teacher will never talk about this, okay? If you go to college, I don't think they're gonna talk about it also as well. So let's take a look. Let's first take a look how they attach, okay, the DNAs here. So this is where the Olympiad is stepping above, like, you know, the step a little bit above the AP level here, okay? I mean, dude, this test, guys, is crazy, by the way. Okay, so this is easy stuff. Okay, it's nothing fancy. So let's take a look. The reason for this is this, okay? So the first thing first, guys, when they're producing a piece of DNA, like when they're with DNA, okay, they gotta put one more, okay? I remember talking about this, okay? So the nucleotide base, the single base they're going there, okay? It's actually what? It's actually ATPs, like ATP, GTP, like CTP, you know, like they're what? So what happens is, let's say for example, guys, the next piece is A that they have to match, okay? It comes in as what, guys? It comes in as ATP. What they call this what? DATP, okay? Because it's deoxy, okay? There's missing an oxygen, right? So what I'm saying is, the basis that you guys were aware of, right, is what? Oh, you have a phosphate group on this, okay? There's what? There's a carbon there. And this is a ribose, and what? That's nitrogenous base, right? Like this is adenine, whatever. We know this, right? So this is what you call as a base, okay? That's what you see on the DNA. But what I'm saying is, before they're being attached, they come as triphosphates. Okay, you know what I'm saying? Which is what you gotta understand. Where do they get the energy from to create this long piece of DNA? Is what? Yeah, of course the ATP. So this being unstable is what? That's what they provide them the energy. And when the DNA polymerase is what? Like putting this guy onto the complementary strand, what happens, guys? They will get rid of the 2 phosphates. We talked about what those were. Like what are they? We call this what, guys? Pyrophosphate, right? Remember this, guys? Adenyl cyclase. Take these 2 guys. What? So pyrophosphate being taken out, and then what? Put it on to the next step. Put it onto the what? The complementary strand. Okay, that's how it works. So why again you cannot produce from the 3 to 5? It's not because of this. Okay? It's not because of like structure of this. So the reason behind this, okay, why this doesn't work the other direction is because of what they call the proofreading. Okay, so keep in mind, guys, the last thing organisms want is a mutation. Okay, which means what? Making mistakes. When they're doing this, okay, they're really fast. Okay, then also what? It's also what? Very, very accurate. Okay, so which means they always proofread. Okay, when they're doing the replication process, very, very accurate, very, 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 really quick. Okay, so but the proofreading, you get what I'm saying, guys? Okay, proofreading. So like you gotta understand, right? So if I sort of zoom in here, okay. So basically, guys, this is a 3-2-5 end, okay, existing piece, okay, and the guy's running. So DNA polymerase is running down, okay. This guy's running down, and produced from the 5 to 3 end, okay. This is like they're producing from the 5 to 3 end, going this direction, okay. Yeah, let's assume. How about I go? Let's assume, right? They made a mistake here. There's a mistake here, okay. What do you think they're gonna do? So as they run the process, they proofreading it, okay? So what will happen is, keep in mind, guys, there's so this DNA polymerase will act as, okay? Keep in mind, guys, okay? Nuclease. Okay, it's called the exonuclease. Exonuclease. Okay, so so there's endonuclease, there's an exonuclease, okay? Exonuclease cuts out the DNA from the end, okay? So if you look at it, right? So which means, dude, okay, like you went this way, 5 to 3 direction for the DNA polymerase. What happens, guys? But like, dude, like you can't start from here. If this guy acts from the 5 to 3, they gotta start from here and delete everything. Okay, so which means what? This guy works what? 3 to 5 direction. Okay, they're gonna just step back and what? Fix it and go back to the 5 to 3. So which means like this guy will go from the 3 to 5 end. So the exonuclease reaction by the DNA polymerase went from the 3 to 5 direction. Okay, so then what? Okay, so they go back. Okay, they move back and what? Fix it and run back down. Okay, that's how the exonuclease work for DNA polymerase. Okay, so why am I saying so? This because of this function. Okay, so that's the reason why they run from 5 to 3 end. The reason is, let me explain. Okay, a little more detail. Let's assume, right? Let's say this is what happens, okay? So this is a DNA, okay? This is basically, guys, typically, let's say at the tip, you got a 3 phosphate there. Okay, this is at the end, okay? This is like the 5 end, whatever, okay? 3 end or 5 end, whatever. So, and then we have a, this is a 1 base, okay? And then this is a 2nd base, sorry. This is 3rd. So I'm just going to go through, okay? So this is what we have, okay? And this is the 5, this is the 3rd 3 end, like a 5 to a 3 end, okay? So now if I explain this, right? If they have another one coming, okay, as per our discussion, right? Okay. So this is the next one, right? They want to add on top of it, right? So which means what? Yeah, dude. So again, so how they attach? Okay, how is this guy going to attach itself there? Okay, so if you want to know, this is what they call in the chemistry perspective. It's what? It's called SN2 reaction. Okay, SN2. Okay, it's a substitution reaction. Okay, so SN2 nucleophilic. I mean, so this is outside of you. This is you want to learn about this, guys? Take Olympiad chemistry. Okay. So basically how it works, okay, is what they got an OH group here, okay, so this will attack the carbon, okay, take out the OH group and what? Take out the 2 phosphates go out. So that's for the energy perspective, okay, what happens is this phosphate will replace the OH group, okay, and what? Make a bond there. That's usually how this works, okay, so that guy goes away. So this is needed for what? Providing the energy, okay, and they take the OH group out and that's basically how it works, okay. But what I'm saying is now, okay, so they put the wrong one here. Okay? Then what happens, guys? What happens, guys? So then they're gonna remove this guy. Okay, this guy will be now removed. The exonuclease will remove this guy. Okay, so we made a mistake. Let's get the hell out. And what happens, guys? Put the right one in there. So this is again, next guy comes in. Okay? And what? Put the right one in there. Do you understand what I'm saying? Okay? It works because what? Like, dude, this one comes as an ATP, right? Like GTP, ATP, whatever. Okay? So yeah, they got the energy. But now the problem, let's do the same thing. Let's assume about the same thing here, okay? So let's say the replication goes from 3 to 5 end. Let's say it's going this direction. Okay, let's say it's going this direction. Okay? So let's see what's gonna happen. So we got a DNA, okay? So let's say we have another base of DNA, right? So this is the one that we're gonna attach stuff onto. So the reaction between this is a 3rd carbon, this is a 5th carbon, okay? Which means what? This guy, so the one that was attached here is the one that will provide the energy because they got 3 phosphates there, okay? And then what? Okay, so this guy will go into a nucleophilic substitution. 2 phosphates on this guy will pop out, okay? And what? Make a bond here. Do you understand what I'm saying? So which means what? Yeah, this is the guy that what? Initiates the process. What? They got the energy here, okay? So now this is what it looks like right now. Okay, do you understand what I'm saying? But now, okay, this is wrong one. This is wrong, okay? Which means what? There is an exonuclease that must what? I mean, let's say assume there's one that goes from the 5 to 3. Okay, it should because they have to go back. Let's say you deleted this guy. Okay, let's get the hell out. Okay, you get the hell out. So this guy is gone now. Problem is what? What now? There's no phosphate here. It's gone. So which means this guy was taken out, okay, by the previous what? Addition, okay? So even if, so this is the reason why we don't have 3 to 5 direction, because what? Even if, okay, we have a DNA, we have what? Correct base now, wants to what? Like attach here, they can't, because why? They don't have a phosphate here. They don't have 2 phosphates, which provide the energy for what? Making a bond here. You understand? That's the reason why it's what? It must be from the 5 to 3 direction, because what? The proofreading perspective, okay? So, I mean, they can do it, but if they made a mistake and have to what? Delete, they can't extend the piece, okay? So that's the most likely the reason why they have to run this extension from the 5 to 3, not 3 to 5, okay? So that is Olympiad, okay? So you probably think, I don't know what you're thinking, by the way, but this is not in the reading. They just, I mean, trust me, okay? If you go to school, okay, and you guys all go to like the Jericho, right? So if you go to school, they will never say, okay, if you raise your hand and say, okay, why is it always 5 to 3? Let's see what they say. You know, like, I mean, I don't know. So, yeah, I'm sure they know, but I'm sure they know what the hell is going on, but it's not in the scope, of course, but it's definitely in the scope of the Olympiad, okay? So if you go to college, you probably have to go to biochem to learn this all, by the way, because it's not in the chem belt. It's not a textbook, okay? Anyhow, so keep that in mind. Like all these things are very high-level chem stuff, so you don't have to know that. It's not even AP Chem, it's organic chem, by the way. So, I mean, you guys took AP Chem, so you probably never heard these things. So that's the reason why they're running down in the existing piece 3 to 5, and what make a complementary them to 5 to 3 for this specific purpose, I guess. Yeah. So they run down and what? Like, yeah, I'm sure you heard, right? Like this guy, the helicase runs down, and this guy runs down, and what? And keep on producing the strand until what? Until it's the end. Yeah, this is the end. Okay. So now, dude, I'm sure you heard, guys. Like this is a problem now. Okay, this guy's a problem. Okay, so because it can only produce from the 5 to a 3 end. Okay. So this side is what they call the leading strand, because it's like pretty nice and quick and smooth, okay? But now this is the problem, right? Because like they have to wait, so which means like yeah, they have to wait until it opens up, okay? Which means, guys, if you look at it here, they gotta put the primase there, okay? Primer there, and then the DNA polymerase will run down this way, okay? This guy will run down like this direction, okay? And then basically what will happen is, so this is a 5 end, this is a 3 end, they're extending it. So, and then, which means what, they have to wait until it opens up again, okay? So which means, I mean, the helicase opens it up again, what happens? Then, yeah, they have to put another primer and what, extend this piece. So it's running down this way. So they'll have various, like these fragments, they'll all have these fragments, what? They have to wait until what, this is completely open, okay? So that's the reason why this is what they call the lagging strand. Okay, and they got these fragments, and these fragments are what they call, guys, like Okazaki fragment. Okay, they're very basic stuff. Okay, you've probably heard those in the Regents classes as well. So, lagging and leading. Okay, it's all resulted of what, guys? The DNA polymerase can only run from the only run and produce the strands from the 5 to a 3 direction only. Okay, we talked about why. Okay. So, I mean, like, dude, okay, so, and then, like, if you look at this side, I mean, so this side, if you look at it, this is a 5 side, that's a 3 side now, okay? So if you look at it here, what will happen is, so this guy will run down smooth, and this guy will be the fragments, you know, so which means, like, later at the end of the day, the next thing is what? There's something they call, okay, so this one is this one, guys, is a different DNA polymerase, okay? So this is what they call, guys, this is a polymerase number 3. There's a DNA polymerase 1, okay? So the next guy is what? So this guy is gonna go in and what? Remove all the primers, okay? So this guy's gonna go into what? Okay, so what? These primers are the RNA pieces, okay? They're gonna go in what? Remove them all. So basically, if you look at what we did here, so I'm going to draw this again. Basically, guys, I mean, what we have here, what we copied looks like this. So this is basically, guys, we made, like, there's a primer here, okay, we made a copy, okay? And then, like, I'm gonna ignore the Okazaki fragment here right now, and then there is a primer at the edge in what? Rundown, okay? So there's like Okazaki fragments, these are the primers, whatever, okay? So basically, guys, what will happen is, okay, these guys right here, polymerase 1 is gonna go there and what? Remove the RNA, puts the DNA, okay? So these primers are now gone. Okay, these primer is gone. So, yeah, this is where the problem is now, okay? And again, no, first thing first, guys, they got something called ligase. So ligase is there to attach the fragments, okay? So like ligase is like to attach them. So this is very basic stuff. Okay? Ligase is there to, like, you know, connect the fragments, okay? But now, yeah, look at the problem, okay? So these are at the end. Okay, these guys are at the end. Okay, they put the primers at the end. Okay, so this is where, like I tell you guys, like where the 5' end is, right? The 5' end here. They have to put the primer there, you know, to extend it, blah blah blah. The problem with this is what? What's the problem, guys? So there's a primer there. What is the problem, guys? If they remove this, okay, so yeah, the DNA polymers wouldn't move this. What is the problem, guys? We talked about, right? DNA polymerase, they can only extend. They can only extend the piece, okay? This was the 5' end here, okay? There's nothing here. So they remove the primers. What happens, guys? They are unable to what? Fill this gap at the end, okay? They cannot fill this gap as what? They only can what? If there's a piece here, they can extend it, okay? But there's nothing there, nothing they can do, okay? So these at the end is what, guys? There's nothing you can do. So what do they do, guys? They cut the DNA. They cut it. So, or just leave it as is. They leave it as is. Okay, so which means, okay, what does it mean, guys? Every time there is a DNA replication, what happens, guys? Every time that gets what? Shorter and shorter. That's basically what it is. Because why? That last piece of the primer, they can't fill it up. Okay, that's the reason why they are getting shorter and shorter and shorter. Okay, so we talked about it, guys. Depends on the length of the edge. So your DNA and my DNA, what? We have something called telomeres. Telomeres, what, guys? A repeated nonsense unit. Okay, so it's like if I'm going to TTA, GGG. Okay, so it's repeated. So at your end of your DNA, end of my DNA, they got telomere. Telomere is a symbol of what? Like, okay, so they got these what? Repeated, you know, TTAGG, TTAGG, TTAGG. That's basically what it says at the end. Okay. So it's what? It's so yeah. We don't use it. Okay. Which means what? Yeah, like it's there, and what? That replicates. Like bye bye. We don't need it. Okay. But there's a problem, guys. It's a limited what? It's a limited amount of telomeres. Okay. So when the cells divide, what happens? Guys? Telomeres getting shorter and shorter and shorter. Okay. What happens then? If there's a gene here, if there's an important gene there, what happens, guys? Okay, so it's getting shorter, 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 shorter. If the piece of gene gets deleted by this, what happens, guys? Cell will know this and what? Kills itself. Okay. The limitation of how many times cells can divide is based on a lot of times the telomeres. So again, like, dude, like, you know, so there's a thing where we talked about, I think last week a little bit. There's a thing where there's an enzyme called telomerase. There's an enzyme. So telomerase, I mean, dude, I tell you, man, like, if there's a way, you know, that every cell in the body can have a telomerase, guys. I mean, I don't know what the implication for this. I don't know if it's good or bad. I'm not sure. But there's a cell in your body. There's a cell in your body that has a telomerase, which is like for the guys, not for the woman, I think. This is for the germ cells, okay, which they can produce sperm cells. Sperm cells can produce, like, dude, they can like divide like how many times they want to, because they got telomerase, okay. Telomerase is what? They extend the pieces, extend the edge, okay. That's what they do, so that what? They can divide more, okay. But the other part of cells, they might have the coding, but they cannot. They don't use it, okay. So. There has been cases where the cancer cells have telomerase. Cancer divide a lot, right? Divide often, uncontrollable dividing of the cancer cells. and they have what? They have a telomerase. So we talked about it, guys. Like future doctors, like you guys must be a future doctors, guys. Okay. So so there's a lot of ways, guys. Like they stop the cancers, the cells to divide. Like one of them as well. One of them we can the cell division, right? The mitotic cell division we talked about. They can hinder production of the microtubules for the cell division, right? So that's one, right? Oh yeah, we can put something that like the cells, not able to divide. In that way, also, yeah, they put a telomerase. Then once they divide a certain amount, they can divide anymore. Okay, so that's one of the ways for the cancer treatment. Okay, the things are getting smarter. Just so you know. So just so you know, but again, it has an impact on all the cells. So which means like if they start to attack telomerase or inhibit them, then yeah, they cannot reproduce. If they die, they cannot reproduce. Okay, so because what? Like the cell probably will not divide anymore, or something like that. Okay, so so a lot of things that you guys have to think about, not just like this is going to now dude okay, so if you guys are gonna be like future doctors or researchers, okay, so what can we do about these things? Okay, I mean I'm too late, guys. Okay, so, I'm just whatever. But you guys are should look at a bigger picture than I am. Okay. So again, like replication of DNA, guys. I mean, this is not the biggest part. Just this is like a very small part of this entire chapter. Okay. But there are certain things you can read by yourself. Okay, in the reading. Okay. But there's certain things like telomerase, like the function, how they work. I'm not gonna talk about it. Okay. But at least the proofreading part, guys, something you have to know. And now the repairing. So they talk about this like on the AP. They talk about this on the Olympiad as well. The repairing of DNA. So they talk about one thing very often, even on the AP, okay, which is skin cancer. We talk about skin cancer a lot, okay. Skin cancer is due to, okay, dude, it's a textbook freaking question, okay. It's due to UV ray, okay. So UV ray, okay, is prone to attack thymines, okay? When the two thymines are together on the DNA strand, okay? When the two thymines are together, okay? And they're attached to adenine. So again, so what will happen is UV ray, okay, you guys all took AP Chem last year as well. Based on the equation E equals HF, they have relatively high frequency, which means the energy is relatively high. This is good enough to cause an electron excitation, okay? Which I'm not gonna go too crazy, okay? So, which means what happens, guys? They can break the hydrogen bonding between A and T, okay, and what? Make them create a covalent bond between the two thymine, okay? That's called thymine dimer. So the carbons on the thymine are now covalently bonded, and in bio, okay, in bio, stupidest thing ever, they think the covalent bond is the strongest thing ever out there, okay? So now they have a thymine dimer, okay, which will be what, guys? Yeah, if they go and start to replicate what happens, guys? Potentially, yeah, this will be what, guys? They'll make what? Mistake in what? This will be a mutation, because we know. So, what's permanent change in DNA? So, what I'm saying is, think about it, guys. DNA replication is what? DNA replication is basically what we call semi-conservative, right? So there's now TTs. What? They're hooked up together. Okay, they're not whatever. So this piece of the strand will make a proper copy. Okay, because what? They have a mutated a copy. But now this one, who the hell knows what they're gonna put there? Okay, they're in thymine dimer now. Okay, do you know what I'm saying out here? Which means what? If a cell has if it's cell replicated, your skin cell replicated, and what? It's got this one. What happens, guys? Yeah, who knows what the DNA will be? Okay, if that portion of the DNA creates some kind of gene, as long as if that portion of the DNA has a gene that produces some kind of protein, yeah, it's not gonna work properly. So, and it can cause cancer potentially. Okay, so that's the main thing I talk about, guys. So what do? They have to repair this, right? Repairing. So this kind of repair is not done by DNA polymerase. Okay? There's a different proteins that fixes it. They call this nuclease. So we talked about it, right? There are many different types of nuclease, but now so we talked about endonuclease, right? I mean, so exonuclease is what the fixed at the end, right? There's something we call endonuclease, which is what this is the one that fixes stuff like this. Okay, during in the middle of the DNA, they excise, they cut this part out, and what properly put the right ones in there. Okay, that's done by endonuclease. Okay. So that's one of the stuff they talk about a lot. The diamine dimer, that's a very popular topic with the UV ray stuff like that. Okay, on the Olympiad, AP, maybe AP, AP now they kind of dumb the questions a little bit and turns into a freaking like essay questions now and reading comprehension now. But they might talk about it, who knows? All right, let's take a look at that, guys. Read the textbook, guys. I mean, this is where I tell you guys, I can't babysit you guys and like, oh yeah, read next to me. I can't do that, okay? Alright, guys. Now, so guys, we gotta talk about time protein synthesis, okay? Okay, so this one will take a little more time. I don't know. Homework, we'll see. All right, so now, guys, it's called protein synthesis. Okay. I'll keep this as short as possible, guys. Okay. So this is where, guys, you guys should know, which is DNA to an RNA. Yeah, to protein. Yeah. So you probably heard this, guys. I mean, so there's an old saying, okay? So the kids be like Watson and Crick, okay? They said this, okay? Like one gene to one protein. Okay, that's what they said. These guys said, okay, so DNA has a gene, okay, which is what turns into protein. One gene, one protein theory, okay? But it's incorrect, of course, guys. By the way. So based on the course of this chapter or this lecture, guys, we'll probably have to understand. This doesn't make sense. Okay, this is not how it is anymore. Okay, they kind of changed it, which is like one gene now like to one like polypeptide. Like, I mean, no. They said, okay, so they said one gene to one enzyme or something. I don't know, enzyme or something. And then they noticed that okay, not everything is enzyme, so they said one gene, one protein. And still, that's not true because what protein can have like many polypeptides, like subunits, guys, by the way. So, like for example, hemoglobin has what, like 4 polypeptides, like as a protein. So they put one gene in one polypeptide. Okay. But this is even not correct. So after end of this chapter, okay, you have to understand this is not even correct. Okay, it's a lot more complicated than what this is. So let's take a look at it. Okay, DNA. So quickly, okay, you got a DNA. So location of the DNA where it can produce a functioning protein or the RNA, that's called a gene, okay? The gene of interest or gene, whatever you call this, okay? This is what they call, guys, a gene. This is where they can actually produce some kind of protein, okay? So this is now 3 to 5, 5 to 3. I'm sorry. This is 3, 5. Okay. So, like, I'm going to be pretty quick, guys, by the way. So as we know, a cell has what, like for our cells, right? We have what? We have a DNA wrapped around in a histone, right? We talked about histone, right? Histone is where our DNA is wrapped around, okay? And our DNA is what, guys, in terms of chromatin? Chromatin is it, okay? There are 2 different forms of chromatin. It's called euchromatin. There's something called heterochromatin. Okay? Euchromatin, heterochromatids. Okay? The difference between a euchromatin to heterochromatin is what? The degree of what? Methylation and acetylation. Okay? We talked about it a little bit last week. Okay? So the degree of what? Methylation. So DNA methylation of what? Histone acetylation. So what I'm saying, what we discussed was this, okay? Not every part of the DNA is needed for a cell. So what I'm saying is, guys, okay, like, you know, like every cell in your body have exactly the same DNA, okay? If there's some cells doesn't have DNA, some cells in your body, they don't have DNA at all, such as what? Red blood cells. Red blood cells don't have DNA. 그냥 DNA 없어요, 걔네들. There's no DNA for them, okay? Keep in mind, red blood cells are erythrocytes. There's no DNA. No DNA means what, guys? There's no protein synthesis. That's what it means. Okay? There's no protein produced. But now, other than that, guys, I mean, they got what? They got DNAs. Okay. But we talked about it a little bit, guys. Yeah. Liver cell. Okay. Liver cells have every single DNA as what? Every other cell in your body. Okay. Because what? Like, dude, cells like has divided, mitosis, blah blah blah, right? Liver cells have what? The gene to create insulin. You understand that? Yeah, insulin. Insulin is what, guys? A hormone that's produced in what? In pancreas for what? To reduce the blood glucose level, right? Liver cells have a DNA that codes for insulin. Okay? Yeah, do liver cells ever produce this? Yeah, liver cells, right? Do they produce this or no? Should they ever produce insulin, guys? They should never. Then what do you think would happen, guys? Then the portion of DNA that has a code for insulin is what? They must what? Heavily what? Methylate and what? And deacetylate that portion of a gene. So a portion like chromatin, which has an insulin coding is what? It'll be very much methylated, CH3. So keep in mind, yeah, this is very obvious, right? When you methylate, yeah, much methyl, guys, CH3, right? CH3. CH3 is what? It's hydrophobic, which means what? Like, dude, water cannot get there, okay? Like, dude, they hate the water, they coagulate, okay? So they methylate the DNA, and also what, guys? The histone, right? Histone is a protein. They deacetylate. So histones have what, guys? Acetylate, acetyl group, right? So acetyl groups have what? Oxygen in there, okay? Oxygen makes them very hydrophilic. So when they deacetyl, they're removing the acetyl group from a protein histone, which makes them further hydrophobic, right? Which makes them what, guys? It's making them heterochromatin, which will what? Like, dude, like even if they want to produce this portion of insulin, what happens, guys? Portion of DNA which codes for insulin, they're not exposed at all. Okay, they're just completely locking it down. So that's what they call, guys, a heterochromatin. Okay, this is all what they call pre-transcription regulation. It's called pre-transcription regulation. So it's before transcription. Transcription is what? DNA to messenger RNA. So like, dude, they can't even let them have a transcription happening, okay? Because why? This is like blocking making that happen, okay? But now, if like, dude, the liver cells are producing what? Liver cells are producing, like we talked about, guys, an enzyme that breaks down glycogen, okay, for example, okay? That must be what? They have to have that portion of a gene exposed, okay? That's what they call, guys, euchromatin. Euchromatin is what, guys? Relatively what? Like demethylated. Their methyl group is just not much there, and what? It's heavily what? Acetylated. The histones are acetylated, okay? And what? Make them more hydrophilic, and what? The methyl groups are demethylated, which means what? Yeah, they make this part a little bit more hydrophilic, make them exposed for the DNA to what? So this portion of a gene to be ready to transcribe. Okay. That's the first part. That's like the first thing, right? Okay, so is it euchromatin, heterochromatin? Like, what the hell is it? Okay. And then, let's take a look at this, okay? So yeah, so okay, so they're ready to produce something. I tell you guys, it's a lot more complicated than what it is. So yeah, they okay, so it's all there, like go take it, Not even, okay. There's more needed for this, okay. Like, dude, they're not gonna just start producing messenger RNA for this, okay. It's a lot more complicated, okay. But we don't. That's regulation chapter, which is next week. I'm not gonna go too crazy right now, but so first thing first, guys. This is a transcription. Okay, we gotta talk about transcription. Transcription again is what? It's from DNA to messenger RNA. So it's similar between similar with the prokaryotic cells and eukaryotic cells, by the way. Okay. Keep in mind, guys. Prokaryotic cells, they can methylate. Bacteria, they can methylate. Okay. But bacteria, they don't have a histone. They got no histones. Okay. Which means that their acetylation and stuff like that is not present in prokaryotic cells. Okay. They can methylate all they want, but not acetylation. So one thing, right? Like one thing fun, right? We talked about this, right? Yeah. I told you, right? 50% of prokaryotic cells die every single day because of viral infection. I'm not kidding, guys. Like, they die almost every day. Okay, because not because of, like, oh, they're old. Like, dude, they die because of the virus. Okay. Yeah. Dude, how do they survive until today? That bacteria, like these prokaryotic cells, they're out there. Okay. Like you get infection, bacterial infection, right? How do they freaking survive? They get injected with the lethal DNA every day. Okay. How do they survive? What do you think, guys? Any idea, guys? Like, okay, so what do? So basically, guys, this is a question for a different day. But once a foreign DNA comes in, right? Like they think it's composed of threat. So they got something like this. Okay, something called restriction. Okay, endonuclease. Okay, they got an enzyme. Okay, called sometimes I don't know if you heard this guy. Your teacher will say like this. Okay, restriction enzyme. Okay, sometimes they call it restriction enzyme, but it's a restriction endonuclease. So they have an enzyme. So prokaryotic cells have an enzyme called this. Okay, once a foreign, like if they think they were infected by a virus, okay, what happens, guys? This enzyme, what does what, guys? It will cut up a DNA. It will cut up any DNA. Okay, it will just chop them up. Okay. So which means what? Okay, so yeah, like, oh, your viral DNA, oh, you came in, oh, come here, let me chop you up, So yeah, they chop it up. Okay, so they what? Possibly what? It's not going to be what? Potent. Okay, but what? Yeah, what do you think the problem? Yeah, what do you think the problem is? Yeah, what do you think the problem? This will cut their own DNA. Okay, so dude, prokaryotic cells have DNA. Okay, they're gonna cut their own DNAs. Okay, so what do you think they're gonna do? What do you think these prokaryotic cells will do? They will start to methylate their DNA, okay? Because that's the only thing they got. They're gonna start to what? Methylate. So they what? So it's like praying that what? This will not get to their DNA and cut their own DNA up, okay? That's their defense mechanism, okay? That's like evolved, to survive. So we use this enzyme. So our human DNA, we don't have stuff like this, okay? It's like we don't have like, oh yeah, COVID virus came in, okay, let's chop our DNA. Like, there's no such thing, okay? We don't have this, but now we use this. We use the restriction enzyme, okay, from the prokaryotic cell in the biotechnology, okay? Something to be discussed. But they don't have like histones. They're not gonna start to acetylate stuff, okay? Okay, so the first part, guys. Okay, so DNA to messenger RNA. Okay, the first is initiation. So initiation, elongation, termination. Okay, that's like basically what initiation, elongation, and termination. So you'll hear this a lot, guys. Okay, right? So okay, this is a genome interest. Like that's the piece that you're gonna try to make a copy. Okay, so that's called a template. This is called a template strand. Okay, it's called a template. And this is just just like complementary strand here, okay? Non-template. So if you look at this, guys, so right outside of the gene, okay, they got something called, okay, it's called promoter. It's called a promoter zone. Okay, promoter. Promoter zone is where like the enzymes will know, oh yeah, this is where the genes that we're trying to look at or try to transcribe as well. So this is where it starts. Okay, I'm sure like I don't know, heard guys, they got something like a TATA box, TATH, repeated TATA. Okay, and then the enzymes will know, oh yeah, this is where it starts. You start. Okay, so. This is, I tell you guys, it's a lot more complicated. I don't want to keep mentioning, waste my time, guys, by the way. So there's a regulations chapter, okay, where they recognize this site and they'll know where it is and stuff like that. So a transcription factor protein called transcription factor, they'll know where this is, they'll bind to it and then try to create a complex and stuff like that. But again, I'm not going to go too far right now. The promoter zone, this is where the RNA polymerase, okay, there's something we call, guys, an RNA polymerase, okay, will bind itself to it, okay, to the promoter zone, and then start to run down, okay, the gene of interest in the template strand and produce a messenger RNA. Okay, so this guy will run down and what? Produce the messenger RNA. Okay, so it's very similar to producing, a DNA replication, by the way. So this guy runs down, okay, produce messenger RNA, and this messenger RNA. So again, like, okay, so it's very simple, guys. Okay, but initiation is what? Yeah, find the TATA box promoter. A lot more to discuss. I mean, exactly how things gonna work. We'll need to discuss this on in the regulation chapter, because they will talk about a transcription factor, like general transcription factors, specific transcription factors in RNA polymerase, like assisting molecules and the what do they call it, guys? Activators and distal control elements and stuff like that. So that's, if I talk about that, guys, I'm not going to finish today. So there's a different chapter I'm going to mention those. But RNA polymerase will go and start to run down this template, okay, and produce messenger RNA from 5 to 3 end. Okay, this guy can only produce from the 5 to 3 end. Okay, they're going to run down and what? Produce to the 5 to 3 direction. Okay, same thing. They're all the same. So elongation, okay, initiation is based on TATA box, but blood elongation is what this guy run down for this 5 to 3 messenger RNA, and termination. Okay, so at the end of this, guys, there's a termination sequence. Okay, they'll know when to end. Termination sequence and what? Like they'll stop this process. Okay, it's very simple. Like they're not going to ask you a lot about the transcription. Like they're simple. They're not too bad, by the way. Okay. So they produce what? Like this is what they call, guys, a pre-messenger RNA. So keep in mind, guys, by the way, okay, this is where it's a little bit different with a prokaryotic cell versus a eukaryotic cell, okay? So this is where the difference comes down to, okay? And once they produce a messenger RNA, for eukaryotic cells, there are more steps to this, okay? So, okay, for a prokaryotic cell, okay, once the messenger RNA is produced, they directly go to the ribosome and start to translate. Okay, there's no like hiccup there. Okay, so a lot of times transcription, okay, which is a messenger RNA production, and translation, which is a protein synthesis, okay, it occurs in tandem for a prokaryotic cell. It comes like it copy and produce protein, and that's it. Okay, and they get rid of messenger RNA right away, and just get over with. So they're very simple, just so you know. But eukaryotic cell is very different, actually. Okay, in terms of how things gonna work. So first thing first, they produce. They finish the production of the messenger RNA. Okay, and now they're gonna start to process this guy. So this is where you guys have to know this. Okay. So like transcription is like this is like you can read this, guys. It's nothing fancy, nothing really to talk about. And now the next piece to understand, guys, is that what the hell do they do with messenger RNA? So, your AP level, okay, your AP level, and Olympiad doesn't really go that far, also as well. 5 to 3 messenger RNA. So guys, what you guys have to know is this, okay? So this is messenger RNA. Within the messenger RNA, okay, there is a portion on the RNA where it should not be translated, okay? And those are what we call the introns, okay? So they have some like in a portion of the 5 ends, okay, they have some like like some here the leading strand like portion, not leading, I'm sorry. This is where guys like they have some UTR zones in the 5 end. It's untranslated region here, okay. So this part they leave it alone, okay. But now here, what will happen is like on the portion here, guys, they have something called the exons, but they have something called introns. And it's a repeat of the exon and introns, okay? Intron, exon, whatever, okay? And then, like, they got 3 ends. So there's some portion that's a UTR zone. We have a 3 ends. So this portion is kind of needed, but they don't really, this part that doesn't get untranslated there. So they have a repeat of exon, intron. So only, you guys probably heard this, only exon portion are the ones that will be translated, which means this is the portion that will be what? Being translated into a protein. Introns will be taken out. Introns are being taken out. They gotta take the introns out, okay? So now, so listen very carefully now, but there's an enzyme, so can I call this enzyme? There is something that will take these guys out. Introns are being, they're gonna cut this out, okay? So introns are being cut out. So what cuts it out is what? Small nuclear RNA. snRNA. Small nuclear RNA. So it's snRNA. Okay? So with bunch of proteins, okay, with a bunch of proteins. So small nuclear RNA is a part, so small nuclear, I memorized this. It's called small nuclear ribonucleic protein. Okay, you found the mistake. Okay, so it's called SNRP. Okay, and yeah, like it's in the video. Okay, it's so basically small nuclear RNA with bunch of proteins, ribonucleic proteins. Okay, nucleoprotein, thing. So so with this guy with a bunch of proteins. Okay, it's called a SNRP. It's something called SNRP. So we talked about this, right? Yeah, what do we call Andy, right? What do we call an RNA that act as an enzyme? What do we call those? So I mentioned this in class, okay? Probably the recordings as well, which I'm sure you go look at, right? So small nuclear RNA act as an enzyme, okay? And these are the guys that actually knows what introns are at, okay? Based on the sequence, they know where it's at and what. Like there's proteins and they're gonna cut those, okay? So what do we call an RNA that can act as an enzyme?
RNAs.
RNAs can act as an enzyme, yes. Catalytic RNAs. What do we call those?
Ribozyme.
Oh, you go show. Oh, right. Okay. So remember this. Okay, these are what? Ribozyme. Very nice. Very nice. It's called ribozymes. Okay, ribozyme is what? It's an RNA that acts as. I'm just messing with you, by the way. Okay, it's like you're my best student, okay. Alright, so RNA, okay, is what? Ribozyme. Ribozyme is what? RNA can act as an enzyme. Okay. So basically, what I'm saying is, okay. So this is a very so this process, right? Like transcription and translation is a very conserved process in a way, okay? Which means, so yeah, RNAs are like old times, RNAs needed to work as an enzyme, and this is what it is, okay? So basically what happens is, guys, like SNRP with a bunch of proteins, like a bunch of more proteins, okay? They turn into something they call spliceosome. Spliceosome is the more it's a complex of all these proteins and small nuclear RNAs. What? And they're the ones that will cut the introns out. They will know the sequence of introns, where they're located, and what? They'll cut these guys out. Okay, and we'll put the exons together only. Okay, they literally cut it out, and we'll cut the we'll what? Now put them together. Okay, so this is where the RNAs will act as RNA acts as what? Like catalytic functions, right? So 5 ends, and now you have UTR zone here, okay, which is like it's just the initial portion of it, and now you have the exons. Okay, so exon 1, exon portion 2, exon portion 3. Okay, now the 3 and UTR. Okay, so okay, they do this every time. For every time we have to transcribe, okay, we need to take the introns out. But you gotta ask yourself, guys, like, what the hell? Why do we do this? Prokaryotic cells, they don't have this, okay? They're much quicker. But now you gotta ask yourself, guys, okay? Like, okay, why? Like, you gotta ask your school teacher as well, okay? When you go to AP Chem, AP Bio, guys, like, ask them why. Like, what the fuck? I mean, so what the hell? Why? Like, why do they have introns? Like, what's the purpose of this? Like, okay, why? Like, okay, so it's a very cumbersome process to take the intron. Like, what the hell? And there's an enzyme that does it. Like, okay, what do we have? Why do our DNAs have this? Okay, what do you think, guys? Like, what makes you think this will help the process? What do you think? How does this help the process? Anyone guys? Yeah. The even AP will ask this. Okay, AP will ask is why do we have introns? Okay, and a lot of students can say like A to Z, like, it can be anything, okay? Let me tell you. One, you gotta be very, this is like, having, Olympiad having this is very, like, I don't know. Very easy, this is easy stuff, by the way. It's called, what we call, guys, alternative splicing. It's called alternative splicing. So, keep in mind, guys, so AP will not make you like these things. I don't think AP will test you on this, okay? But you gotta know what this is, okay? They don't care about like, oh, do you know what cooks it like? But you gotta know what this is. So keep in mind, guys, like the Olympiad will test you what I said, okay? Small nuclear RNA will know the sequence for the UTRs. I mean, so for the intron zone, and they'll know where to cut. Okay, that's what they'll ask. But you know, these things, I mean, of course, Olympiad, they will make you know. But now, what this is this, okay? Remember what I said? Yeah, listen carefully, okay? One gene, okay? One polypeptide, okay? This is incorrect because of what? Because of this. So basically, what I'm saying is this. So a lot of times, guys, these exons, right? Exon 1, 2, 3. These will become, so once this actually gets translated, okay, these portions will become the domain of a protein. Okay, so what I'm saying is domain is what? Domain is like location of protein. So what I'm saying is, guys, this is a protein, okay? This is a protein. Like protein looks like whatever, okay? Right? Like this looks like garbage, right? So, yeah, so what I'm saying is, guys, so E1, okay, is this part of the protein. Okay, E2 is like this part of the protein. Okay, this is the second domain. So the first domain, second domain, is the third domain. Okay, so what I'm saying is, guys, okay, so this is the exon portion 1, 2, 3 is what? Yeah, it can result in what? First and second and third part of the domain of the protein. Okay, so protein has like, you know, some parts here. So like, for example, what? Okay, so this is like a membrane protein, for example. Okay, like a cell membrane protein, for example. Okay. But then, yeah, this is what? These are like a protein channel, for example, whatever, okay? And then this is what? Something that can act as a catalytic function on the cytoplasmic portion of the, like, whatever, okay? So these have a separate functions, potentially as a protein themselves, okay? But now, what this is, alternative splicing. This is what it is. When they cut, when the introns, when the small nuclear RNA, the spliceosome is cutting out the introns, depending upon the signaling perspective, what happens, guys? They can do this. They can cut from here to what? Like, so which means like they can cut the exons out as well. So which means what I'm saying is, yeah, like this is something they can do. Instead of what? Instead of, guys, E1, 2, 3, it's what? It could be what? E1 and what? Just E3. Okay? You understand what I'm saying? So the second exon portion is what? It's gone. You don't have a second exon, okay? Because what? They cut that part out. So which means what? If they produce a protein like this, if they translate this messenger RNA, what happens, guys? The second domain is gone. So when they produce protein, what happens, guys? It's a different protein. It only has what? 2 domains, 1 and 3. Okay, domains of the protein. This guy's not there anymore. Why? Because that's what they asked. That's what the signaling probably said, okay? So that is what they call alternative splicing, okay? Based on how the spliceosome cuts up the introns and exons, okay, when they're processing, it can result in what? They can result in into a different protein with different domains, okay, present, or, you know, like one like thereof, whatever. That's what they call as alternative splicing. So which means, well, yeah, it can be a whole different protein, different functions, and stuff like that. Questions, maybe? This is one reason. Okay, this is one reason of it. The second reason is, people probably heard this, guys. I mean, they don't explain this very well in the textbook as well. It's called exon shuffling. And I don't know if we can talk about it, by the way. It's called exon shuffling. So exon shuffling, what it is, like people like they're like they think like they're crazy, man. Like they think, oh yeah, you can put this here, you shuffle the like, no, that's not what it means, okay? Exon shuffling, this means this, okay? So what it means is, so we haven't talked about it yet. This is something to discuss during meiosis, okay? Meiosis occurs within the germ cells, like sperm cells and egg cells, blah, 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 okay? That's where the exon shuffling occurs, okay, during meiosis. So one of the big thing about meiosis is what? You have two homologous chromosomes, okay? Like two chromosomes that are homologous. They're not the same chromosome, okay? They're not the same, but they're what, guys? They're like, it can impact the same traits, okay? Like meaning, guys, like the hair color. So this, you got one from your mom, one from your dad, okay? Like, yeah, like, okay, so, which means they're not the same thing, okay? They're not the same. So, what happens is, like, during the meiosis, what happens, guys? There's something like crossing over. They talk about this a lot, man, on the AP. They love the crossing over. I don't know what the hell, why, I have no clue, okay? They love crossing over, okay? Especially in this chapter. So what happens, guys? Yeah, they can this piece and that piece are what? Exchanging, okay? So yeah, now what happens, guys? Yeah, this is now, you know, like exchanged. Okay, so yeah, so now some parts will have a gene from this side, and they're swapped, and which further what improves the recombination of DNA. Okay, like yeah, that's the reason why like you don't exactly look like your dad. Okay, that's why something like this happened. Okay, like I don't know. Do you want to look like your dad, guys? Like exactly, like exact copy? I don't know. I don't know you guys. I don't. So but I don't know. But that's what the hell is exon shuffling? So because in your DNA, there's an intron portion, right? There's an intron portion, okay? Because of there's introns, the exon portion, so the DNA that will become an exon portion of messenger RNA, they're more spread out, okay? So exons are not like squeezed in, they're like spread out, okay? So which means, if you look at a DNA perspective, okay? Okay, so there's an exon here, an exon there. What happens, guys? They could have been swapped, okay? So having the intron portion on the DNA, so DNA which can be an intron, it what? It kind of like extends out or separates the exons, which will make sure what? Yeah, there could be more crossing over occurs, this is what they call exon shuffling, okay? Which is what they're shuffling exons from one portion of a DNA to the other portion of DNA during crossing over, okay? That's what exon shuffling is. And there's a better chance of shuffling because of the introns, right? They're spread out, right? The third piece, mutation. So what it means is mutation is what? If there is a mutation, okay? Like for example, guys, like just like we talked about, like dimer mutation from the UV rays, right? Like okay, so there's more introns than exons, by the way, in the DNA. Okay, there's like DNA way more introns than exons. Okay, so which means like okay, so there's something happened. Okay, you like you got like people like these white people, they like this. I mean, so to some people, okay, they want to go out and tan. Okay, and like why? I have no idea. But like if they get tanned, like I get tan easily. Also, I get these farmers tan. I get very dark. I don't know why. Just from the driving from the car. So So, I mean, I hope I'm not a farmer. Otherwise, I'll be talking like Spanish today. I mean, just but, no, guys. Okay. Oh, hablo español, right? I'm sorry. I love my, my friends next door, down south a little bit, but I'm sorry. But, well, 맞지? 네, 무슨 말이다 맞지? The UV rays timing, right? So, but let's say, for example, guys, DNA occurred. So DNA occurred, guys, The mutation occurred in the introns. What happens, guys? What happens? Yeah, what happens, guys? Does it have an impact, guys? Absolutely not. Okay, which means, well, yeah, dude, like, yeah, those are something we call, guys, what? Yeah, silent mutation. Okay, like, yeah, it might not have impact at all. Okay, so yeah, having introns, which is, well, yeah, if there's a mutation, it might be really bad for the prokaryotic cells, by the way, because what? Everything gets copied, turns into protein, but not for us. Okay, so if something happened in the intron portion, it might not have an impact at all, for the functioning perspective, okay, for the proteins. So those are some of the benefits of having introns. I mean, what's the downside? I don't think so. I mean, downside is very cumbersome. It's slow, And it's a lot more parts, okay? So like, for example, guys, if you guys like have a computer, like super like complicated computer, it's more prone to break. If something is gonna break, guys, I mean, yeah, like, because there's more parts. It's hard to fix. All right, so let's take a look. So basically, so the introns are being taken out by this enzyme, and so there's more to it, right? So the exons are there. I mean, we talked about the exons. Depends on what the exon gets there, guys. It is what it is. And now, on the 5' end, they put actually what they call GTP cap on this guy. So there's a cap, the 5' cap, the GTP. So which means they put a guanine, okay, cap with like 3 phosphates on them, okay? So on the 5' end, guys, they have a guanine. Guanosine triphosphate, okay, GTP, right? So on the 5' end, and then also, guys, on the 3' end, there's a classic one. They put, so this is AP, okay? They'll ask you guys, what happens at the 3' end, guys? They put what? They put something we call polyatal, okay? So they put like like long adenines on the 3' end side, okay? There's a name for this. I mean, what do they call this? Adenylation or something? That's the name. Adenylation. It means like they put the adenosines on them. Adenine on them. Adenosine. Yeah, it's just A. The purpose of this is what? To make sure that what? Like, yeah. So these are all of this right now is happening in the nucleus. Okay. But once this guy goes out to cytoplasm, there's enzymes. There's what? Like, yeah. There could be a lysosome, which what? Which, if you put a polyatal, what happens then? Yeah, this will protect the degradation of the messenger RNA. Okay, that's your answer. If they ask you, okay, what the hell is the purpose of polyatal? It's what? It's what? To prevent degradation of the messenger RNA. That's the same thing for the 5' end GTP cap as well. Okay, 5' end GTP cap. That's the 5' end GTP cap. It'll become one of the signal for binding onto the ribosome, okay? But other than that, this is also what act as what? Preventing degradation as well, okay? So that's something you can read in the textbook, okay? It's very easy stuff in the AP level. So now like, the process messenger RNA is ready, they will go out to the cytoplasm for the ribosome for the translation process, okay? Let's take a look at, guys, translation now. Okay, translation. So, keep in mind, guys, for your information, so for how we talked about this in the regular class, right, the AP class for you guys, and I didn't really mention it too much for you guys. So how you guys, so on the AP side, don't get confused on this one. I mean, if you really know it, you can kind of mention this on the AP as well. So, first thing first, okay? The initiation of a translation, okay, occurs in cytoplasm, okay? So what I'm saying is, ribosome is not something that it's not like always bound to the ER. That's what I'm saying. So what I'm saying is, so initiation always occur in the cytoplasm, okay? So the initiation of the translation always occur in the cytoplasm, okay? What I'm saying is, the location of the ribosome, we say, oh yeah, there's one in ER, there's one in cytoplasm, so they know, okay, so if the enzyme will be used, the protein will be used in the cytoplasm, it goes to that guy, but if it's something that will occur in the ER, like, it will be excretion protein, then it will go to ER, the rough ER. Now, that's not how it works, okay? So that's just for the dumb class, you know. So what will happen is, so ribosomes, as we discussed, there are two subunits, right? There are smaller subunit, there's a larger subunit, okay? So what will happen is, if this happens in the cytoplasm, okay, it doesn't happen in the like they don't know yet, okay? So what will happen is the messenger RNA from the 5 to a 3 end, okay? So this is a 5-inch deep cap there, polyethylene. I'm not going to mention it like 5,000 times here, but now this will bind to, okay, smaller subunit ribosome first, okay? This will bind to it first, which means this is what will happen now. The 5 end will be binding to the ribosome, the smaller subunits. So, so what will happen is now, this is important, guys, okay? So basically, guys, so this translation process has an initiation, there's an initiation, okay, there's an elongation, termination, same thing, okay, initiation, elongation, termination. So the initiation is, okay, so first thing first, messenger RNA binds to the smallest subunit ribosome, okay, in the cytoplasm. And now, every time, so you guys, let's talk about this, okay, so you guys are not like dumb kids, okay? Just so you know, think about it, right? There are 20 amino acids. So, dude, last year I was explaining this to kids. They absolutely they had no clue what I'm saying, okay? And like that's AP kids. Amino acids, 20 amino acids, and they're like, oh my god, like I can't believe it. At 20 amino acids, okay? So you gotta understand, okay? We got 20 amino acids. Somehow the coding on the messenger RNA, okay, should what? Relate to a specific amino acid. Because what, dude, whatever the coding that went on the messenger RNA should what? Code for a specific amino acid. There are 20 amino acids, okay? And we got 4 different bases, okay? These are single-strand messenger RNA, single-stranded, okay? So what I'm saying is, yeah, think about it, guys. Can a single base on the messenger RNA, okay, like whatever this can be what? This can be like A, G, C, U, right? Could be any one of these guys. And we talked about why, like I'm not gonna go to, so can a single base on the messenger RNA can specifically code for an amino acid, like, which means like, okay, so if it's A as a base, I'll put this amino acid, specific amino acid. If it's a G, put the different amino acid C. There are 20. It doesn't make sense, guys. It doesn't make sense. What, dude? I mean, you can do up to 4 amino acids for a specific amino acid. What about the other 16? It doesn't make sense, right? So what about, okay, so okay. So what if we make 2 sequence of the 2? Okay, so it could be also A, G, C, U. Okay. So which means what? Like, what's the maximum combination can we get? We can get AA, like AG, AC, AU, okay, GA, GG, C, like, you know, so we can as well, which means what? We can get up to what? 16 different combinations of the code, okay? Is this sufficient to represent 20 amino acids? It's not, okay? Which means what, guys? We need what? Triplet codes. So we need three of these bases. So we need at least 3 base coding, okay, to represent an amino acid. Okay, so yeah, we need what? Okay, so A, G, C, U. Okay, so the 3 of the coding, 3 base coding should represent 1 amino acid. Okay, we call this what, guys? On the amino acid, on the messenger RNA, we call this what, guys? A codon. It's called a codon sequence. Okay. So keep in mind, guys, I mean, so like how many unique combinations can we have, guys? 64. It's a lot, guys. 64 for 20 amino acids. Okay? So which means, so what happens is, what will happen is what, guys? Which means that 2 different codon sequence can code for the same amino acids. So which means that because of this, and look at this, okay? For example, like if I'm missing a UCU, UCA, UCC, UCG. Okay, these are the messenger RNA triplet codon sequence. Okay, yeah, these all, so like what I'm saying is if the codon is this, okay, all four of them, they all code for exactly the same amino acid, which is what? Serine. Okay, so I mean, typically the first two are the one that really matters. The first two, like really dictates what it is, but you know, it's the last one. A lot of times it doesn't matter. Okay, so which means what? So this is so there's a name for this. This is what they call wobble. Actually, wobble. You gotta know the name, guys. Wobble is what? Yeah, the on the codon, the third sequence of the codon. Okay. Like it's fine, okay. It doesn't make a big difference with the amino acid because what? Yeah, like it does bring a difference, but not so much, okay. So usually the first two are the ones that really matters, and the last one, like whatever, you know, like it does come into a factor, but sometimes it doesn't. Like for example, like the amino acid serine. So, which is the codon sequence of the messenger RNA is this, or this, or this. It codes for all seven amino acid. Okay. So based on the codon sequence, right? But now what you have to understand is this: on every, this is like, dude, this is for every translation process. There is something we call the start codon, okay, which is what? AUG. There's always, guys, AUG. This is what they call a start codon. So on every translation, this is where they start, AUG, okay? So which means it doesn't matter if you're prokaryotes or eukaryotes, okay? This is a start codon. That's when they start, okay? That's a start sign. So there's a UTR zone when they're gonna see AUG. Without AUG, it's not gonna happen, okay? So with the AUG portion, which is start codon, so they're gonna put a transfer RNA. So now it's a transfer RNA. So tRNA, okay, so which is the matching tRNA is what, guys? This is a UAC. Okay, UAC transfer RNA will be bound to a specific amino acid, which is a methionine. So methionine is always the first amino acid. Okay. So at the end of the day, once the translation is over, the first amino acid is always methionine. Okay. So this is messenger RNA on the bottom. This is basically what they call transfer RNA, tRNA. Okay, with a specific amino acid right now. So keep in mind, guys, you have to know this for AP as well. Okay, where there is an enzyme. Of course, there's an enzyme. It's called amino acid synthetase. Okay, synthetase. Okay, this is enzyme. This enzyme, okay, there are 20 different amino acids, 20 different proteins like this that work, guys. This is the enzyme which will bind a specific amino acid to a specific sequence of a transfer RNA. If a transfer RNA sequencing is like UAC, okay, then what happens, guys? This enzyme will what? Put like methionine on them. Okay, this is a big this is a enzyme catalyzed process of amino acid onto the transfer RNA. Okay. So this is keep in mind this happens at the expense of ATP as well. So without the ATP, it's not gonna happen. Okay. So read this in the textbook. Okay. Nothing fancy. So right here, so this is a smaller subunit. Okay, that's a larger subunit. It has the, I'm not larger. I'm sorry. This is basically the initiation. You know, where the start codon, the first one right there. So this is the initiation part. Okay, this is the initiation part, and also what? There's a larger subunit here. Okay, and the larger subunit will be there. Bind to it's a larger subunit as well. Larger subunit, guys, has I think I mentioned this to you guys already. EPA zone, EPA, okay, on the larger subunit. So E portion represents the exit, okay. They represent exit. P stands for peptidyl, okay, peptidyl. This is amino acid, okay. That's what they stand for, I think, if I'm not mistaken. So basically what happens is, guys, so this guy right here, so will be on, so now this is a larger subunit here, and what will happen is this is E, this is P, this is A, okay? So right now, this right here is the end of the initiation complex for translation, okay? So they need to have the smaller subunit and the methionine with the UAC transfer RNA. So this is how the initiation for the translation occurs, okay? And then, this is interesting now. So keep in mind, guys, just so you know, like binding of a messenger RNA onto the ribosomes. How the hell does this occur? It's what? The ribosome has what? Ribosomal RNA. So our RNA act as a ribozyme, okay, will be able to determine the messenger RNA and what, and then helps or assist of binding of the messenger RNA onto the small subunit, okay? So there's a function of the RNA as well, guys. Yeah, dude, it helps assist. There's a catalytic function of these RNAs, okay, on the rRNA as well. So now we're gonna start to discuss now the elongation, okay, of the amino acids now. So basically, guys, I'm gonna raise this now. So this is what it looks like, okay? Smaller subunit, guys, a larger subunit. I'm sorry, this messenger RNA. A larger subunit, okay, EPA. So we have guys one here. So this is AUG, and now we have a methionine here, okay. So what will happen is, guys, so the next guy with different amino acid, with the same, whatever, okay. So this guy will be going into the first phase, guys. I found the missing, guys, is there's a name for this. It's amino. The codon, maybe the first one. There's a step I forgot. I mean, it's a codon recognition. So if I'm not mistaken, okay, it's a codon recognition. So which means, what it means is what, like this guy. So this, whatever this next coding is, whatever, like so UC like A, whatever, okay. So it'll be like the codon and anti-codon recognition. So this guy will be what, like A, G, whatever, U. So this guy, the transfer RNA will go into the next one. Okay. So again, there's a ribosomal RNA has worked as an enzyme within the ribosome as well. Okay. This guy will be going in and binds to it. Okay. So this guy will bind to this part now. Okay. So this guy will be going in. AUG is gonna go. Now this guy has the amino acid there. That's the first part, the codon recognition. And second part is basically what they call a peptide bond formation. Okay, peptide bond formation. So basically what will happen is, guys, here, so basically what will happen is, so again, they have these ribosomal RNA, okay, which will function as like, the enzymes and stuff like that, right? So basically what will happen is, they'll put this guy, so basically what will happen is, guys, they'll break this, okay, and put this guy on here. So basically at the end of the day, what will happen is, they'll break this part, and they'll actually make it into like this, okay? So they will elongate portion here, okay? So basically, they'll take the portion here, okay? They'll make a peptide bond with the existing and the new amino acids, okay? And then they'll put it onto the transfer RNA on the A zone here, okay? This is EPA. That's basically what they call creating a peptide bond between existing to new amino acids. And third one is what they call, guys, a translocation. There's a lot of the term translocation. Translocation term, guys, by the way, is applied also in a couple chapters, by the way. So translocation, basically what it means is this will be now, these two will be moved, okay, to the next portion of the rest. So this guy will be now going here, this guy is moving here, okay? It's gonna move, like, so they're gonna, so you can think of it as what? They're running down, or like this is moving here. So, which means now, so this guy, which is which is the first transfer RNA, will be moving to the E zone, and this guy is moving to the P zone, okay? And now what? The next next transfer RNA is ready to come in and what? Try to like match the next messenger RNA. So that's called the translocation. Okay, so this will continue. This will continue on, and what? Will elongate the amino acid chain. Okay. Until when, guys? So now we have termination. So this is elongation, right? So now termination. Termination is what? So termination is based on the messenger RNA coding. So messenger RNA coding is what, guys? So there is something we call the end codon. This will tell you what, when to finish, when to end the translation. Okay. Either the sequence of the end codon is what, guys? UGA, UAG, or UAA. So there are 3 codon sequence. That's what they consider as end codon. Okay, so that's, you gotta memorize them up. They're not gonna tell you. AP, they will tell you. Okay, AP, but oh yeah, go ahead. This is end codon. This is a start codon. They will tell you. Olympiad, they will not tell you what's a start or end codon. You gotta memorize them. So I don't think they're gonna go that far. So I don't think they're gonna go that far of like you have to memorize all these like it's triplet codon. Like I don't think they go that far. Okay, but I mean again, I can't say the same for the amino acids, by the way. But again. Like, okay, so these guys at least know these 3, 4, okay, at least know those. End codon. This is what? This will tell you what? Stop the process. Okay, stop the process. Okay. So once they read this portion of the codon, what happens, guys? What will happen is water will go in and what? This will actually hydrolyze the peptide. Peptide goes out, okay, and then they will disassemble. Okay, messenger RNA goes out. Okay, they will all separate now. The smaller and larger end of the ribosome will separate as well. So keep in mind, guys, by the way, so this is what they call, guys, a translation process, right? But now, you gotta understand, guys, okay? During the translation, okay, this isn't the reading, okay? This is like nothing fancy, okay? But I don't think this is the scope of the AP. So during the translation. So basically, what will happen is, the process of initiation and elongation, okay, it starts in the cytoplasm. For any translation, it starts in the cytoplasm, all of them, okay. But how do they know? Like, how do they know? Okay, it's supposed to be secreting protein. How do they know that? So meaning, guys, okay, so they are doing all this, okay. They're producing the protein, okay? They're producing the long chain. So they have produced, okay, this much, like long chain of amino acid, right? So keep in mind, okay, there is something called signal recognition protein, SRP, signal recognition protein, okay, in the cytoplasm, if I'm not mistaken. Did I write this? Yeah, signal recognition protein, yeah, SRP, yes. So there's something called signal recognition protein, right? So they will be able to read off. So there is a signal here. So they will, based on the, not on the messenger RNA, but based on the amino acid sequence, okay? There's a protein that will read, okay, able to tell if this protein is meant to be used inside the cytoplasm or this is a secreting protein, okay? If they realize, okay, this is a secreting protein, then what happens then? That protein will bring the ribosome into the rough ER. So this is ER, and what? They will have this thing bound to the rough ER and what? Have the polypeptide produced in the lumen of the rough ER, okay? That's the determination of what? By the amino acid sequenced by the SRP, okay, which is a signal recognition protein. Okay, based on the amino acid sequence, they can tell whether this protein is meant to be used for the cytoplasm or to be going out, and this guy will assist to binding this into what? The rough ER, okay, and further produce further produce amino acid and as a translation inside the lumen of what? The rough ER, okay? That's translation, okay, nothing fancy, okay? Yes. So your homework is I assigned it, it'll be enough to solve questions with this information, okay? So, all right, let's take a look at your homework guys, by the way. So whatever you did today,